The overall goals of the following experiments are to understand the properties of excitable membranes and the ionic basis of the resting membrane potential, and to learn methods to measure the membrane potential, as well as to demonstrate properties of synaptic transmission. In one part of the experiment, the resting membrane potential will be measured while altering extracellular potassium. In another part of the experiment, mechanisms of synaptic differentiation will be investigated by recording stimulated synaptic responses in various motor units. Next, a neural circuit is presented that is easy to maintain. This preparation can be used for teaching as well as researching various aspects of a sensory-to-CNS-to-motor neuron-to-muscle circuit. This set of experiments show the ease of using the Crayfish model to address issues relating to membrane potential, synaptic integration and transmission, and structural differences of muscle fiber types.
The main advantages of using the Crayfish preparation in teaching electrophysiological concepts and techniques are the ease of dissection, the preparation's viability of minimal saline, the ease of obtaining synaptic responses, and the ease of discerning the overall anatomical arrangement of the muscles.
These techniques can also be applied to other preparations and will further our understanding of synaptic physiology and modulation and biochemical and structural differences in the demonstrated preparation.
To begin the surgery, select a crayfish approximately 6 to 10 centimeters in body length and place it in crushed ice for 5 minutes to anesthetize it. To decapitate the crayfish, hold it from the back of the head, making sure to keep your fingers out of reach of the crayfish's claws and mouth. Using large scissors, quickly remove the head by making a clean cut from behind the eyes of the crayfish. Remove the legs and claws of the crayfish to avoid injury. Remove the stylets on